How's that? Can you all see that on Zoom?
Yeah, this looks good.
Perfect. Okay, great. Well, uh, I'll start by passing this around. For people on Zoom, I brought some pinned lanternfly, but you don't get to enjoy the amazing experience of looking at dead bugs. But if you all want to pass this around, you're welcome to. It has their life stages from first instar when they first hatch all the way up to adulthood with the dates they're in nature. But thanks for having me. Really excited to be here. So let's see if this will actually move.
This key.
Yep. Okay, great. So just briefly, I'm gonna go over the outline. So I'll just give a brief scientific background about myself, go over spotted lanternfly as an invasive plant pest here in the United States, go over spotted lanternfly's relationship with tree of heaven. There are only two acronyms in this whole presentation. There's SLF and TOH, spotted lanternfly, tree of heaven. So if you can get those straight, we'll be in good shape. Going to talk about my research, characterizing and quantifying that relationship, and then also future directions and broader impacts of this research. So I'm from Delaware, first state, went to college at Gettysburg in Pennsylvania. And while there, I got to work with an ecologist down at the Smithsonian Tropical Research Institute on a number of different animals, and that really cemented my love for ecology. And so I got to work on this multi-trophic interaction between these two frogs, these midges that like to bite them, and also bats that like to eat them. And there's a laser pointer on here, right? That'd be great to use. There we go. Okay. Also got to work on tortoise beetles and predator-prey interactions with them as well. So ecology is great. Try to hide these things so y'all can see it better. Okay. So I did that, got back to the United States and graduated. Then I got to work up at an AREC in Winchester on this invasive brown marmorated stink bug and these parasitoids that lay their eggs in the stink bug eggs. So it's what we would consider biological control. I got to work on a different system, emerald ash borer, as well as the parasitoid wasps that parasitize their larvae and their eggs. Yeah.
So, those are parasitoids. Are those parasitoids native or non-native?
They're non-native. Yeah, they co-evolved with the BMSB over in Asia. But yeah, so I got to work in ecology, tropical ecology, and then I got to come up here and work on invasives. And so now I get to work on another invasive doing chemical ecology. And so that's what I'll be talking about today, the spotted lanternfly. They are a plant hopper, and so they have piercing sucking mouth parts that allow them to feed on tree sap. They're originally from Asia, China, Southeast Asia, parts of India. Now they're here. They're currently present in 20 US states. They originally came here in Berks County, or they originally came to Berks County, Pennsylvania in 2014. And since then they've radiated out. So 9 or 10 years, they've spread quite far. And if you look at the I-81 corridor, they've actually spread from a satellite population in Winchester down to Southwest Virginia. We get a little bit more local. They are not here yet. So this is an iNaturalist map. They're not in Blacksburg, but they are in surrounding areas. There's quite a lot in Roanoke, and there's quite a lot in Radford. I think it's only a matter of time before they do come to town. In terms of why we actually care about them, wow, that formatting got really screwed up. That's okay. When they first emerge, they are black and white. Young nymphs are black and white, and they emerge in late spring, early summer. When they do emerge, they're going to feed through the new growth of a very wide variety of different plant species. And then as they age, you'll see that in their fourth instar, their penultimate life stage before adulthood, they'll get this bright red coloration, and then eventually they'll become adults. Once they do become fourth instars and adults, they can actually pierce through bark to access phloem and xylem that way. They also get this host preference shift. Their hosts sort of narrow in as they get older. And so they really like to focus in on tree of heaven at this stage, as well as some other hardwoods, and in terms of, economic importance, grapevines. And then, of course, they will mate to round out their life cycle and lay eggs. If y'all haven't been out of town or if you haven't seen their egg masses, this is what they look like. It's kind of a smattering here. Up here and then down here in this corner, you'll see what they look like when they have this protective proteinaceous egg covering over them, and that's to protect them from temperature changes and humidity and predators. You might see them without that covering and they're just laid in lines. And then there's also sometimes old ones with holes. You can see that they hatched the previous year. And so they're kind of cryptic. You might have to look a little hard to find them, but when you see them, you'll know what they are. And then since this is the Virginia Native Plant Society, I figured I'd compile a list of natives and non-native invasives. So on the left, it's all these native plants. And then on the right, there's non-native plants as well as, cultivars that people like to plant. They also really enjoy feeding on these highlighted ones when they reach adulthood. And so for native plants, they feed on these hardwoods like maples and black walnut, staghorn sumac, even the native grapevines that we have here, like Vitis labrusca and Riparia. In terms of the invasives, they really love tree of heaven, bittersweet, chinaberry, Chinese sumac, and then of course the cultivated varieties of wine grapes. So there's quite a wide variety here. They'll lay their eggs on even more things: native trees, invasive trees, stop signs, rail cars, trucks. And that's one of the reasons they're spreading so well. They like to hitchhike with our man-made objects. Consequences of their feeding, you can get an oozing trunk wound like this, especially if they're feeding en masse. The plant might experience wilting. There could be branch death or plant dieback. It might reduce the overwintering capacity of plants to, survive till following season. If you're growing things like caneberries or wine grapes, it can reduce your fruit yield over time. And then, of course, if you have a lot of them feeding consistently, especially over multiple years without treating them, they can kill your plants outright, but that's pretty rare. Indirect effects of their feeding are really just honeydew. And so honeydew is their liquid excrement. They're feeding on liquid all day, so they're producing a lot of liquid excrement all day. It's very sticky. So if you're ever under a tree where a lot of them are, you might feel rain. That is their poop. On that honeydew, a lot of sooty mold will often grow, and that in turn will reduce photosynthetic yield for foliage underneath where it grows. It can also be unappealing if you're, in horticulture or agro-tourism.
Does the honeydew attract other insects?
It does, yeah. Research came out recently that it attracts other lanternflies. It also attracts stinging insects and flies, bees, right? Collecting these things, you go down on the ground, there's, wasps kind of fighting with you. So yeah, really attracts insects that like sugar. There can also be a foul odor from fermentation if you're ever around these things. You can kind of see here, there's some white coloration that's the native bacteria and fungi doing fermentation. So it smells a lot like vinegar if you ever find one of these heavily infested areas. So it's a very well defended pest overall. It's great at eating, great at escaping predators like us. So it can close its wings and hide against things like tree bark pretty well. Can obviously hop very rapidly, as is its name. He can actually play dead, and so I rear these things in a quarantine lab, and they've tricked me before. They've tricked my undergraduates before. Think they're dead, but they're not. They also have this beautiful aposematism, so these bright red warning wings saying "Don't eat me," and dimatism is just rapidly displaying those to sort of shock a predator into thinking, "Oh, I shouldn't mess with that bug." But they're also very well defended chemically, and so that's what I'm really excited to talk to you all about. And that brings in Tree of Heaven. So I'm sure all of you know Ailanthus altissima. It's in every continent except Antarctica, and you'll know it from, side of the highway. It's a very aggressive and well-defended tree, so it has a slew of phytochemicals in it that it uses for allelopathy, so, damaging other trees' roots and stealing their nutrients. It's also well defended against insects and some browsers. I've seen deer eat it, but they eat anything. And so in that large slew of phytochemicals, they have this one really prominent one called ailanthone. And so they actually synthesize that in their roots and they shoot it out into the soil, and that kills other plants around it, allowing it to shoot up more tubers or suckers, I guess. And they also use it for anti-herbivore defense, but spotted lanternfly is special because it's co-evolved with tree of heaven, and it can actually sequester this compound and use it for its own anti-predator defense, which I think is really cool. And so sequestration is really just the ability of any animal to accumulate and store toxins in the environment or any compound in the environment and use it to its advantage somehow. It's usually used for defense. I've seen it used for sexual reproduction as well. And then also there's detoxification. So if an herbivore encounters, a toxic chemical, it can sequester it, but it can also try to get rid of it after it's eaten it. It can metabolize it, break it down into different smaller compounds. It can add different moieties onto it. Also, it can metabolize it further or excrete it out. So there's a variety of things an insect can do if it encounters a chemical like this. And so really this has led me to ask a lot of questions about sequestration. We first want to see if we can detect it using our own methodologies ourselves. There's other publications out there that show that it does provide anti-predator defense. There's been studies with birds. There was a study in Korea that looked at the relationship between tree of heaven and lanternfly, but none so far in the US. So we want to see if we can actually detect it and quantify it in tree of heaven, what it's feeding on, in the insect itself, and then also in the insect's honeydew. And so that led me to a hypothesis that sequestration is occurring if the concentration of ailanthone in the phloem is a lot higher than the concentration in the honeydew, right? They must be doing something, maybe accumulating it over time, if that is the case. And then if that is the case, how efficient is that process, right? By what factor are they accumulating ailanthone? What factor is the concentration changing in the phloem versus in the honeydew? And so to answer that, wow, that got messed up too. I went up to a field site in southeastern Pennsylvania. This was last, I guess, 2 summers ago now. Found a bunch of different tree of heaven. And for each one of those tree of heaven, I made sure there were a lot of different spotted lanternfly feeding on them. And so for each lanternfly I found, I would collect a 3-part sample, some phloem that they were ingesting, the insect itself, and then also the honeydew that they were excreting. And I did that 42 times, collecting a bunch from all these different trees. But today I'm just going to show you a subset of 9 samples just from one tree, because analyzing these things takes a long time. To do that, first I had to collect honeydew, and so this part was pretty easy. Essentially took a cap of a tube and gave it a little toilet. And over time, they use it, and sometimes they're shy, or sometimes my arm gets tired, so I built a little shelf. And they'll still go. They usually don't move. And you get quite a lot. It's 50 microliters from just one go. And that's really all we need to be able to analyze. Getting the phloem was harder though. And so I had to use this technique called stylectomy, where you go up while they're feeding and snip off their stylet while it's inserted into the tree. Because the phloem is under positive pressure, the phloem will exude out of the stylet, and then I can go in and absorb it with a capillary tube and then analyze it later. So it looks like that, nice little globule. And after I'm done torturing them, I freeze them on dry ice, so I don't just leave them to slowly die with no mouth. Bring them back to the lab and then analyze all 3 sample types. So once we have all those samples, to extract them and then run them through the machine we use to quantify ilanthone. First, we'll freeze dry the liquid samples and then resuspend them in a methanol and water solution. For the insects, we actually freeze dry them and then crush them in liquid nitrogen and then extract them using methanol. Once we have all those different samples, we'll normalize them against a background, sort of like giving them all an equal baseline so we can really be sure about how much ilanthone is in them. And then we use liquid chromatography, mass spectrometry. Basically, we're asking how much ilanthone is in this sample per microliter or milligram, etc. The liquid chromatography just separates out the chemicals. The mass spectrometry detects the chemicals, and then we can quantify from there. And this is the one that didn't show up. So this is going to show my results. So I will just skip that and talk about it in a conclusion slide as if I had already talked about it. So, yes, it is detectable and quantifiable. The people we work with at Virginia Tech, we were able to find it and find accurate levels of it. This hypothesis, however, was not that simple. So I was going to show a graph with phloem, the insect, and the honeydew. There's quite a lot of ilanthone in the phloem. There was a little bit less in the insect, and then a very, very minimal amount in honeydew. And so that's telling us they're able to sequester it a little bit, but it might not be that simple, right? It wasn't like I hypothesized, right? They're not sequestering it in numbers or in a concentration that's higher than what's in the tree, but they are accumulating it somehow. They're also likely detoxifying it though, since there's so little in the honeydew. And so they're definitely able to modify it in some way in order to excrete it. It certainly doesn't affect them in terms of living. They do very well eating tree of heaven. And in terms of how efficient is the sequestration process, comparing these numbers, there's 563 times more ilanthone in the phloem than in the honeydew. So that again is just telling us they're doing something with it. They're metabolizing it, they're sequestering it, but we need to learn more about it. And so that's one of my projects in my PhD. Another one is really figuring out how long they can keep it in their bodies once they've actually fed on it. And so I hypothesized that if I would go out and collect spotted lanternfly from mature tree of heaven in the field, that if I just stuck half in cages with grapes and half in cages with tree of heaven, the ones that I stuck with grapes, their levels of ilanthone would decrease a lot faster than those I kept on tree of heaven. I also assumed in this project that lanternfly that I just grabbed out of the field would have really high levels of ilanthone, and we'll see if I was right on that or not. And so to figure this out, again, summer of 2023, went out and collected a bunch of SLF, up in West Virginia. And so we found 6 different mature tree of heaven in the area, collected a bunch. And then for each one of those adult trees, we would put half in cages with grapevines and then half in cages with tree of heaven, replicated that 6 times. And then very simply, we would just collect and flash freeze them days 0 up to 2 weeks to see how well they would maintain the chemical over time. Yeah. And so we use the same methodology, freeze drying the SLF, crushing them in liquid nitrogen, extracting them, normalizing them, and then trying to quantify the amount of ilanthone in the insects. And this graph actually came up. Okay, so on the left, or on the y-axis, you'll see the amount of ilanthone. This is in peak area. It's not a concentration, but it is still relative to what we're looking at. On the x-axis is time in days. And so if we look at days 1 through 3 on just the grapes, there's no significant difference between the amount of ilanthone just grabbing them right off the tree and analyzing them immediately. So it looks like they're able to maintain it over 3 days. If we move on to the insects I kept on tree of heaven, seems like they're able to accumulate a lot more. And so inadvertently, It's able to make them sequester a lot more ilanthone by forcing them to just feed on tree of heaven instead of being able to eat whatever they want in the environment. And then days 7 through 14, I have not yet analyzed, but this is interesting, and I really want to figure out what's going on in the latter half of the days. And so if we bring it back to the hypotheses, we can see that SLF kept with grapevine actually maintained similar levels, at least up to 3 days compared to what they had right off of Tree of Heaven in the field. And then with this assumption that, SLF will have high levels if I grab them right off the field, or right off of Tree of Heaven in the field, that wasn't supported either. They actually have fairly low levels compared to if I just wanted to force them to eat Tree of Heaven the whole time. Moving forward for future research, analyzing the remainder of those samples. That's priority number one. I would like to graduate. Furthermore, determining actually which organs are used for sequestration. A lot of insects don't just put it in their bloodstream, their hemolymph. They will devote certain organs to keeping those chemicals. A lot of them keep them in their cuticles as well. Imagine if a bird is trying to eat you, and I mean, your exoskeleton is the first thing it's going to taste. So I've done a lot of dissections, and I'm looking into analyzing them in the same way. Also looking into determining which genes are actually used for sequestration and detoxification, and that can really help us understand more about their relationship with Tree of Heaven and other host plants that they might encounter toxins in. And that really leads me to this question. This research has really created a pipeline to do this with other plants and understand how they deal with secondary or specialized metabolites as well as primary metabolites in their different host plants. We know they feed on black walnut. Juglone is the compound that's used for allelopathy. It's also very aggressive, just like Tree of Heaven, but it's native. There are plenty of compounds in wine grapes. There are plenty of compounds in bittersweet and all these other plants that they'll feed on. We just don't really understand how they interact with them. Are they also sequestering them? Are they detoxifying them? What's going on? In terms of broader impacts, we hope that this research will enhance our understanding of their relationship. They co-evolved together in Asia. They are both here now. Lanternfly definitely do very well when they feed on them. They don't need them to survive and reproduce, but they always seem to grow faster when they have it. And of course, they're very well defended when they do feed on it. Digging into that more, I think it'll help us manage both of them better. A lot of people tend to ignore Tree of Heaven and just want to focus on lanternfly, but Tree of Heaven actually supports a lot of other invasive insects like ground worm and stink bug, for example. Understanding the Tree of Heaven nutritional content for these insects would be really interesting. What sugars do they need to survive? Why is Tree of Heaven such a good host for them versus something like grape or black walnut? And finally, there's tons of bacteria everywhere, and of course there are bacteria in the vascular tissue of plants, the xylem and the phloem. How are they interacting with the bacteria in the microbiome of the spotted lanternfly gut? Really want to look into that as well. And then I hope this can inform area-wide management. Managing spotted lanternfly and Tree of Heaven is really important that everyone cares, especially in an agricultural standpoint, like where I do a lot of research. Tree of Heaven spreads very easily. The mature trees can produce tons of seeds every year. Spotted lanternfly can fly, right? They can hop and move very readily. And so if you are a landowner and you choose not to care about managing Tree of Heaven or spotted lanternfly, that just gives the vineyard owner down the road really more of an issue later. So it's all connected, and there's actually a grant that will hopefully get funded soon about trying to get area-wide management of both of these underway up in Winchester to start, and then further down in Virginia later. And then this research also just adds to our understanding of how insects use these specialized metabolites. There's a big consortium of people that are interested in sequestration, just so we can know more about it, because it's cool. I think that's really important. Inherent knowledge and whatnot. So with that, I'd really like to acknowledge all the people that have helped me with this research, my advisor, Dorothea Toll, and the Toll Lab, Invasive Species Collaborative, and Translational Plant Sciences Center at Tech, the Mass Spec Incubator that helped me analyze all these samples, my collaborators up at the USDA, and then my committee members here in Biological Sciences and Entomology. And yeah, thank you very much. Taking the questions. Metabolite. Mm-hmm.
Hey, Brian,
Yeah.
can you repeat the question so that we can hear too?
Yes. Can I define metabolite? And metabolite is really any compound that an organism produces. So I was talking about ilanthone in Tree of Heaven. That's a metabolite. I don't know if, have you guys heard about primary and secondary metabolism in plants? Does that ring a bell? Okay. Primary is like anything that a plant uses directly for growth. So like photosynthesis and sugar transport and stuff like that. But secondary metabolism or specialized metabolism is anything that doesn't directly deal with growth. So like defense against herbivores or like sexual reproduction, stuff like that. Mhm. Yeah.
In tracking other insects, they've had traps. Are there traps to track the spotted worm?
Yeah, there's quite a lot of different kinds of traps. Most recently, these guys up in Massachusetts they took roofing shingles and made a cone out of them and then attached it to a tree. And for some reason, lanternfly really like going up in there and laying their eggs. And so that's a great monitoring tool to see if they're actually in the area. Like back in 2018, when they first got to Virginia, there was a lot of use of sticky bands, just glue on paper that you would staple to trees. And that was pretty good for seeing if they were there, but it mostly just caught a lot of bycatch like ants and sometimes birds and lizards, and we don't want that to happen. So yeah, there's those. There's a circle trap as well, but yeah, all sorts of different things. Yeah.
Is anybody studying the predation of the lanternflies?
Yeah, there have been a couple of studies.
Hey, Brian.
Yes, sorry. I had just been talking about different kinds of traps for monitoring them. There's ways.
We can hear your answers, but we can't hear when people ask questions.
Gotcha. Okay, yeah. So talking about ways to, or was it predation of them? Looking into predation. Yes, so there's someone up at Penn State. There was another group a couple years ago looking at different arthropods that would eat them. So spiders and mantids, assassin bugs will eat them. Birds will eat them, but again, if they've fed on Tree of Heaven quite a lot, it will make the birds vomit. So, yeah.
That was really the question, is the level of the lantern in them
Yeah.
affecting predation.
That would be a great next step, because like once you figure out exactly how much is in them,
Repeat the question.
he just wanted to know about man, I've never done this. Sorry. Seeing how much ilanthone is necessary for causing a bird to not eat the insect. And yeah, go ahead.
Do they have any like native Asian predators on like their native lands or?
Yeah, so I asked if there are any Asian predators that would eat them where they evolved. And I imagine there are. I don't really know. I mean, there are plenty of spiders and mantises, and there are parasitoids over there too, actually, wasps that will lay their eggs in the nymphs and in the eggs. And there are people up at the University of Delaware doing biological control studies on them, trying to see if they could approve those wasps for release. But a lot of work goes into that. They have to make sure they won't actually parasitize our native plant hoppers or anything similar.
When, this came in through customs.
Right, right.
So this is expensive. So why are they not being some way fumigated or radiated, or they're in containers. These are things that are products brought in containers. I'm sure, I mean, it's a massive amount of containers,
Yeah,
but it seems like to me it might be, if there was some way to do that, it would be more cost effective than letting all these invasives come in.
yeah. So she asked, she was referring to how lanternfly got here originally, which was likely on a shipment of Oriental stone. The egg masses came in and they hatched, and now we're here. Why wouldn't it be more cost effective to just, fumigate initially? And it's hard to find all of them. You have to train workers to be able to identify them.
And the eggs, it wouldn't make any difference. Eggs probably wouldn't be affected by fumigation, right?
Depends on what you're using. If you use something organic like a myco-insecticide, like, fungi that will destroy the nymphs once they hatch. It depends on when you apply it. For certain insecticides, it won't actually pass through the egg.
That would presume you already know what you're looking for to stop the spread of.
Right.
Rather than individual, like, I'm only gonna stop these, that's not the way you can do it, because then something else gets in.
Yeah, yeah. I mean, I'm not too certain about all of the different, protocols that USDA APHIS, for example, uses to, monitor for their presence. But also a lot of people don't care. They just do not, like, even if they're trained to have to look for them, they, rail workers don't care.
So, it's probably new, but it's gonna have to keep happening. There's no, I don't see how.
Right, right. It is definitely going to keep happening. And so we typically try to look for, management tactics rather than just pure eradication or control. To try to eradicate them, that would have been a problem to do, or that would have been a thing to try in, 2015, 16 when they first got here. Now it's really more about managing their spread, slowing their spread. Yeah.
So if you're out and about, so you said they're not here at Blacksburg, but if you're out and about and you do see the spotted lanternfly in an area where it hasn't been reported, what's a good way of reporting that it's spread to a new area?
Yeah, so what's a good way to report spotted lanternfly if you've seen it in an area that it is not currently known to be in? Stopslf.org would be a great place to go to. For Virginia, you can call VDAX and report it there. I think you can report it online too. They
Pretty much stopped looking for it.
don't care that much anymore. I did get a call back like a year ago once, so that was nice. But
Well, it's so widespread now, you're not gonna stop it, so.
yeah, it's good to know where they are, especially for, agriculture. You can know, spray preemptively if you have a vineyard or something like that.
Well,
Yeah.
I remember the stink bug, that's whatever that stink bug was, it was ABX stink bug. It was like, you did your windows everywhere, it was everywhere, and now I don't notice it as much.
Right.
So, when they first come, like they basically have a boom and then maybe it has its own cell breaking, I mean, why do we not see as many stink bugs? Or maybe now I'll be really sorry.
So she's referencing the brown marmorated stink bug and the kind of boom bust cycle of invasive insects, and why I haven't really seen a lot of those stink bugs. That's very typical of invasive insects. They'll come and they mate rapidly and expand rapidly. Lanternfly's been doing that too. People where I'm from in Delaware say, well, why haven't I been seeing spotted lanternfly anymore? Are they gone? I'm like, no, they just move on. They're really good at sensing which trees have a lot of nutrients that they want. Sometimes we'll see them all just gather on one tree. We call that a hot tree. We're not really sure why. Could be sugars or other compounds. But eventually, yeah, predators will get a taste for them, realize that they're not scary or that they are edible. Parasitoids might try to parasitize them. There's a lot of different factors. Entomopathogens like Bavaria bassiana, I don't know if you've heard of that. It's a fungus that kills insects. There are native strains here that will feed on them. And also just people learning to manage them better. So,
And parasitoids, are all parasitoids specific to particular specific insects? I mean, species?
Mhm.
Or are there generalist parasitoids?
yeah, she's asking about if all parasitoids are generalists or if they're specialists, and yeah, it's a mixture of both. A lot of them are specialists, and usually the specialists are the very tiny ones. There are parasitoids like sawflies, if you've heard of them. They'll just, stick their ovipositor right through bark and lay their eggs in like a beetle larva. But usually they're very specialized, and the only ones that they'll actually release here that are from somewhere else, like Asia or Europe, have to be very specialized.
Yeah, because after you said that, I thought, oh, yeah, they're specialists, but not all of them are. Because that makes good sense. That seems less threatening to me than if it's a specialist parasitoid.
Right.
Even if you don't know, because things can shift to, if it's a parasitoid for a specific genus, then that's problematic, but if it's for a species, that's not.
Yeah, a good example is with brown marmorated stink bug. The samurai wasp accidentally got released here somehow, and that's a specialist on brown marmorated stink bug. There's no evidence that that is the direct reason why there aren't as many now, but they were released somehow, and now there aren't as many of them. So that's cool. We haven't noticed.
People are doing this, know what they're get out. Okay, never mind.
Yeah. Wasp terrorism.
Hey, Brian. Mary asked in the chat, does the ilanthum get passed on to the young?
Yeah, that's a great question. So
What is it? Repeat the question.
yeah, goes both ways. Wanted to know if the ilanthone gets passed on to their young. And yeah, so someone at Penn State was analyzing the egg masses and the first instars, and actually found that they could find it there. So she reared them on both Tree of Heaven and grape. Grapes don't produce ilanthone, and so she found that the first instars still had some ilanthone in them, and it actually dipped as they got older, and they eventually lost it. But the ones that fed on Tree of Heaven just kept accumulating. Yeah.
In your first study, you mentioned you had 42 samples, and I think in both you talked about normalization of that data that you collected.
Yeah.
Could you talk more about that normalization process? I was wondering what kind of variation you were seeing.
Yeah. So if I'm trying to, she was asking more about the normalization process in, normalizing all these different samples together to get a baseline. When you're trying to generate a concentration for something, you need, amount of ilanthone as the numerator and then something as the denominator. And with these studies, the milligrams of insect fresh weight is usually the denominator. And that's, great for getting a concentration, but for actually trying to figure out what or how much ilanthone there is in a context of all the other chemicals there are, you need some other factor, really to figure out what other chemicals are in there that could be interacting with the signal that ilanthone produces. So as all these different compounds are spread apart, they come up to this detector, and so the detector makes a peak for each one that it finds. And that peak for ilanthone might be larger or smaller depending on other chemicals in that sample that will interact with it. And so to normalize them, I had to go through and find an absorbance value. And so I had to dilute all my samples based on that. And then I ran them through the chromatography. It's not fun. Yeah, but does that make sense?
I have another question. In the beginning, you showed, I think it was an iNaturalist map that was sort of zoomed in on our area. And you said it's really concentrated in Roanoke and Radford. It's going to be coming to Christiansburg and Blacksburg. Do you have any hypothesis about why it's skipped us so far? Like, are there more plants in these areas that they like, or something else?
Yeah, she's wondering she's wondering why there are more lanternfly, in Radford and Roanoke, and they haven't been found in Christiansburg and Blacksburg yet. And I really think that's because of the human mediated transportation, right? 81 is what, 15 minutes away from here, and so there's no major, trucks coming into town and stopping like there are in like Roanoke and Radford and Pulaski. So if they are laying their eggs on tractor trailers or on, rail cars, they have time to hatch and then infest the plants in the area. Like Blacksburg has plenty of tree of heaven and plenty of grapes and maples, everything. So they would probably do well. I thought about the elevation, but I don't think that really matters.
It must be a railroad, because
Yeah.
stopping Roanoke and they're like side car tracks and Radford.
Hmm.
Plenty of Christiansburg too, though.
No, not like, no, I don't think so, not like that Radford and Roanoke.
Well, that's true, they have yards there
Yeah.
for layover.
Yeah, I figure if they're stopping there for a while, give them plenty of time to hatch.
Yeah, I had wondered because I think it was iNaturalist data, right? If there was some sort of link also to maybe people are using more iNaturalist in Radford or Roanoke, or
It could be. Yeah. Wondering if people use iNaturalist more in like Roanoke or Radford. It could be it. I don't really know. I mean, there are enough.
It's not, I'm in Christiansburg.
I figured people would be finding them, but
where the parks or something.
I use it to actually collect their egg masses, and so I'll cross-reference iNaturalist with where rail depots are, and I'll just go there and be like, oh yeah, that's a tree of heaven. There's a bunch, and so I'll scrape them off. And, yeah.
So, looking at the insects that you handed out earlier on, do they have spotted lanternfly as like one, life cycle throughout the year? Or do they have multiple generations through the summer?
Yeah, he's asking about how many generations there are per year, and it's just one. There was fear that as they would move down into the Carolinas and the warmer areas that they might become bivoltine, produce twice, but we haven't seen that yet. We don't think it's going to happen. So yeah, it's just once per year.
There's another question in the chat. Do you keep track of the age of adults you analyze?
Do I keep track of the age of the adults? Is that?
Yeah.
Yeah. The question was, do I keep track of the age of the adults that I analyze? And yeah, I do. And that's really important for, well, let's see, there's like two distinct periods when they're adults, pre-mating and then post-mating. If you've ever seen them in late September, October, they'll get this bright yellow coloration on the sides of their body, and that's usually them accumulating fat for laying eggs, or it's getting colder, they want to produce more fat. And so yeah, with like the early September, I'll make a note of what the day that I captured them on. Obviously I don't know when they molted in the field, but yeah, I'll keep track of that.
Which stage survives the winter?
The egg masses survive the winter.
Okay, good. You're scaring me then. Okay.
Yeah. When it freezes, they'll usually actually have their stylet tapped into the tree and they'll just die hanging on the tree. Yeah, doing what they love.
Right, it's amazing.
Do you like this bug or do you hate this bug?
I mean, I think it's a really cool insect. I mean, like thinking, oh, this bug is evil, like that's asinine, right? It's like it's a bug. But I do think killing it is effective, especially on a small scale. Like if you see a lot of them and they're affecting the plants you planted or, trees in a park, I think that it really does actually reduce their population within that area. I think, thinking they're evil and killing them to try to eradicate them from the US is never going to happen and is foolish. But yeah, so I like the bug. I like
Yeah.
it.
Did you just vacuum them?
Yeah.
Rosemary and I had our first encounter in Harrisonburg, Virginia, but you could just vacuum them, right?
And she was asking if you can use a bug vacuum to suck them up, and yeah, you can do that. I use that in the quarantine lab. The nymphs are really small, and sometimes you can crush them by accident, trying to pick them up, so you can just suck them up. Yeah. Fly swatters are good. Yeah. Gotta be quick though. They can't hop backwards. I'll say that. They cannot hop backwards.
Another one says they above that.
Um,
Thinking about a info version and seeing like it's midnight, like zappers.
Just gonna go through to see if there are any other questions in the chat. I have some overwintering in my house. Oh my god.
The stink bugs, Brian.
Oh, okay. Okay. All right. Yeah, that's all the questions, I think. Anything else here? Yeah. No, I took my classes there, but I'm in the biology department actually. I'm in Latham.
I want to ask, maybe I'll learn to. We did it one time before, we met at the Mexican restaurant, and I should have thought about it, but maybe the next meeting we can try to meet it, if people are interested that want to, meet at that Mexican restaurant for the meeting.
All right.
Yeah, wherever that's.
Yeah, Beth here at the, in the room had suggested that we would have a meetup before the meeting next month at El Rodeo, where we could all sit down and have dinner together before coming over to the meeting. So we will announce that for the February meeting. And for the people here from the Master Naturalists, the neat part is we're probably gonna have a joint meeting in March.
We have to be officially approved.
Oh, yes.
So.
Yes, we're waiting. There's going to be that official approval I've just been reminded of. Because the Native Plants Society has been willing to do it. And so we're just waiting for formal approval from the Master Naturalists for the March meeting, which will be pretty good. As far as I know, we had a amazing presentation again by Brian here, and we're very thankful for that. Does anybody have anything else they'd like to bring up before we conclude tonight's meeting? Yes,
Can I talk to the folks on Zoom as well about the Native Plant Propagation Center? I just want
Sure.
to make sure everyone.
Please
heard
do.
a little bit
Yeah.
about- okay.
Emma would like to talk to you. I'm going to see if I can. Yeah, okay. I've got to see if I can actually see the Zoom screen.
Great, thank you.
There we go. All right. Hi, folks on Zoom. I just wanted to make sure I was able to talk with y'all a little bit about the Native Plant Propagation Center at the Blacksburg Community Gardens. I am looking to partner with an organization or a volunteer group or something to actually use this Native Plant Propagation Center that was put in last year with a grant from the Regional Commission and Plant Southwest Virginia Natives. And so I would really love to, both the people in the room and the people on Zoom, if y'all want to reach out and have a project you think would be applicable. The Propagation Center has potting tables, a shade cloth, raised beds for seed starting in like a raised bed area. We've also got established in-ground beds of native plants that are good landscaping types like mountain mint and you know a large variety of native plants. They're slipping my mind right now, but if anyone has relevant projects they think would be good for this, I would love to hear from you and kind of throw some ideas around. I'm hoping to get it started in the next month or two, as we need to cold stratify some seedlings or some seeds probably. So you all can reach me at my email is gardens@liveworkeatgrow.org. That's gardens@liveworkeatgrow.org. And I've got business cards for people in person. But yeah, I really look forward to working with you all and seeing you all around. I'm also very passionate about native plants, so I think it's a good collaborative opportunity. Thank you.
Great, I appreciate it.
That maintains the native plants at the Montgomery Museum.
Yeah, we have already, I already told her all about that.
We had a brief introduction, but I'd love to talk to you more about that.
All right.
And there's a master naturalist pollinator garden at.
So for those on the line, they're having a discussion about the resources that Emma can reach right with the people that are here in the room. All right. Does anybody have any other issues that they'd like to bring up this evening? All right. Well, thank you very much for attending. And we'll see you next month.
So what you do.